Hello and good afternoon. My name is Bjørn Klem. I'm the CEO of AdjuTec Pharma from Oslo. And I'll be happy to take you through the travel of AdjuTec Pharma. And this is a spin-off from the University of Oslo. And Professor Pål Rongved is in the audience. He started this project 10 years ago. And I'm sure he will be happy to take any questions from you afterwards, if needed. Antibiotic resistance is a devastating problem globally, as you all know. There's a great unmet medical need in this area. And I'll tell you a little bit about the patent protected technology that resulted in APC 148 that is now phase 1 ready. As we see from the map, the parts in Europe, in the south and east, are particularly vulnerable to this antibiotic resistance. And we also in the Nordics have encountered this problem recently by all the migrants coming from Eastern Europe that will be war victims or for other reasons, people migrating to Europe. This is taken from an publication during summer, and shows you exactly what I mentioned about doctors in Scandinavia facing a terrible situation, having these migrants coming with extensive bacteria resistance towards all the antibiotics we normally use in this part of the world. And I'd like just to show you also this clip from a paper a few weeks back. A friend of mine from India, he came home with this article and told me that India is actually suffering more than any country in the world from antibiotic resistance, and also towards the most powerful antibiotics we have on the market. The situation is devastating, as more than 50% of all patients in the hospital are resistant to these last sort antibiotics. So how does AdjuTec Pharma help in this situation? Well, bacteria defend themselves by producing enzymes that destroy antibiotics. So what we do, we are producing enzyme inhibitors. So these are not new antibiotics. Actually, APC 148 has no antibiotic effect at all, but it kills or destroys or inactivates, as you like, the enzymes. So together, they are a marvelous combination that will sort out the bacteria, also those producing these enzymes. And just as a reminder, we are now talking about patients in hospitals eligible for treatment of intravenous antibiotic treatment for their complicated infections, being urinary tract infections, lung infections, intra-abdominal, or even sepsis. This is just a cartoon made by Professor Rongved. Microbiologists hate it, but we like it because it's simple. And it explains that in gram-negative bacteria, you see there are two types of enzymes, serine beta-lactamases and metallo-beta-lactamases. For metallo-beta-lactamases, there are no actually enzyme inhibitors on the market at all. And I think this is what triggered Professor Rongved at one time to look if we can find some inhibitors to deal with that problem. For serine beta-lactamases, those enzymes, they have been there are products on the market, including avibactam, as shown on the picture. And we would also like to combine our technology, our patent technology, APC 148, with avibactam, which is available, together with last sort antibiotics, to be able to inhibit all these enzymes all together. Now, this is also very attractive among big pharma companies. They have important antibiotics in the market, but they face increasing antibiotic resistance, and they also see patent expire on their products. So by including this technology into their products, they will be able to keep revenue going and also to extend their patent life. And we see here several examples of deals made in the past 4 years that shows you how these companies acquire or license in this kind of technology that you will hear more about now. This is one of my favorite slides because it's simple and it really explains what we're doing. meropenem is a last resort antibiotic, and it should work below 8 microgram per mil. However, some of these bacteria, they produce these enzymes, as we mentioned, the MBLs, and you can see you need more than 500 microgram per mil dose of meropenem to kill this particular bacteria. So it's a terribly resistant bacteria. You cannot give it to the patient. The patient will die and it's of no use, too toxic. However, if you combine it with our enzyme inhibitor, you see a dramatic shift. So now the bacteria is sensitive to meropenem at very low doses of 2 microgram per mil. So now we're in the range of being able to use it in the patient. And we have done this experiment in many bacteria, resistant bacteria, and it works pretty much all the same. And this is very encouraging as a product for countering the antibiotic resistance we see on a global scale. We also wanted to introduce, as I mentioned, an SBL inhibitor. So actually by having 2 enzyme inhibitors and a last resort antibiotic, we are now able to inhibit antibiotic resistance whether they produce MBLs or SBL type of enzymes. So a broad spectrum inhibitor product. And this is interesting to Big Pharma because they have these products, maybe including the SBL inhibitor, but they lack our technology. So what we do now, we are testing a lot of products from Merck, from Pfizer, from Shionogi, from others, GSK, and see if we can improve them with our technology. And I think this, and we see very many positive results in this testing, and are very eager to go to the companies and say, you got a good product, but we can make it better. As an example, you will see here that this is a product that's meant for IV use. We will administer intravenously and in the hospital, and typically treatment period here will be 7 to 14 days. We have obviously been doing our homework with respect to regulatory. We've been to FDA, discussing it thoroughly with them. We have now qualified for their qualified infectious disease program, granting us priority review. We have option to get fast track and also rolling review on our application for an NDA. We have talked to European Medicinal Agency, and we have cleared the pathway with our first protocol, phase 1, with MPA in Sweden. So we're all ready to go, and we have set up the protocol and a contract with the company to take care of this phase 1 trial next year. Now this is a snapshot of our development program. In green you see the phase 1/2 program. It will include about 140 volunteers and some patients. This is basically safety. We know the product works. This is all about safety, and we will do dose escalating studies, single dose, multiple dose with our drug substance alone, but also with the combination with the antibiotic. We estimate that it will have a cost about $20 million. In today's market, that's a difficult task. So what we are aiming for now first is to raise $5 million and then add on next year with the remaining investment. As you see also, there is a phase 3 program at the end of the journey here. We hope that we will, after the phase 2 program, that we will have so much good safety data and efficacy data that we will strike a deal with the global pharma distributor. We also, of course, are looking for non-dilutive funding. In this area, it's very important, and we also have many good opportunities. As the previous speaker mentioned, they had received the EIC grant. We are applying for it now, and hopefully we can get it. We had a very positive first stage evaluation from all evaluators, and I think the area AMR is of interest to politicians and also all the institutions. So we definitely hope that we can get this grant by the end of the year. Just to give you an impression of what the market is for these products, you see here some antibiotics, some best-selling antibiotics on the market. These are carbapenems and cephalosporins, and typically they sell for about $400 to $500 million per year. We definitely have looked at the competitors in the market, They are few, and you see in the gray shade, there are very few that are handling the MBL enzymes. Most of these are SBL inhibitors. A few of them are MBL inhibitors. So I think AdjuTec Pharma is in a very good position to take a leading position with our technology when reaching the market. We're a small team, only 5 people. 4 of us have a very long experience with drug development from different companies, international companies like GE Healthcare, AstraZeneca, but also Norwegian biotechs. We've been running global clinical programs with many products, so we know how to handle this. But of course, we depend on a very efficient and a good advisory team, and we have put together excellent people here that cover most areas, like toxicology. We have an former FDA reviewer on our consultancy team. We have microbiologists, infection medicine people, and not the least, we have people working on business development. And I dare say that David Finlay is a very valuable asset to us because he has been a former head of GSK Antibiotics. And of course, that is a knowledge that we really appreciate. He knows what our partners would like to see on the other side of the table when we get there. Also, we have a very competent board of directors. And again, our recent person on the board, Tom Berken from Sweden, has great experience from making deals with companies from his position in Amgen and Takeda. And I think this would be a very, very valuable knowledge to have when we start talking to these companies. So to sum up, we know the product works. We have shown it in a number of bacterial strains. We know it's safe. We have done our homework in the preclinical package. We have defined the dose to move on into the phase 1 study. We have sorted it out with the regulatory authorities. We have a clear pathway to the market. And we also see great attraction from the big pharma industry with this kind of technology to protect their assets and get their revenue going for another few years. So with these words, thank you for your attention, and I'll be happy to take any question. And Pål sits right here, so he will also join you if you have questions to him. Thank you.